and welcome to the LA Venture Podcast. This is Minnie Ingersoll, host of the podcast and partner at 10110. 10110 is a seed stage fund here in LA. All opinions expressed on this show by me and my guests are solely our own. Today's episode of LA Venture is sponsored by SaaSoft, offering customized software development for your startup's needs. SaaSoft is a group of top software developers, all based here in Los Angeles. If you are looking to build a new product from scratch, or if you're a founder that needs help accelerating product development, SaaSoft can help. With SaaSoft, you'll get the personal attention you need to make your product a reality in 2021. Head to saasoft.com, SaaSoft, for a free consultation. I'm excited for my discussion with Sean Harper today. Sean is a managing director at Westlake Village BioPartners. Westlake Bio recently raised a $500 million Fund 2 to continue their work funding transformative life science companies. Sean founded Westlake at the end of 2018 with Beth Seidenberg after leaving his role as head of R&D at Amgen. Sean, thanks for coming on the show.
Thanks for having me.
Of course. I'd love to start with the basics. Maybe you could tell me about Westlake Bio and what sort of companies you're funding and what stage those companies are.
Fundamentally, we're involved in company formations, so company incubations, if you will. We're typically putting seed funds in just to get things rolling, hire team, license assets out of universities, et cetera. And then we often are the lead on the Series A investments. And then we try and move these companies through successive rounds of private funding.
When you're doing that first investment, what size check are you usually writing as a first check and maybe a second check?
Our seed funds, typically we start with $1 million. Sometimes we have to add a bit more before we can get to a Series A. On average, it takes 9 to 12 months for us to go from hiring in an entrepreneur or beginning to assess a technology at a university and getting to a Series A. And in that year, typically the $1 million seed's adequate, sometimes a little bit more. And then check sizes for the Series A with respect to what Westlake puts in range from 8 to 35 million.
And just to clarify, those checks that you're writing are always into companies who are working on new drugs. Is that right?
Great question. So yeah, we're very single-mindedly focused on human therapeutics. We have a requirement that there's at least a lead product that is no more than 3 years from IND, from first in human testing. So as opposed to some investors who will invest where the first product may not get into man for 5 or 10 years, we're pretty focused on getting human therapeutics into the clinic, at least the lead program. And I would say that we focus on that because it's our expertise, right? Mm-hmm. This is where we've all spent our time in the industry. So we don't do devices or diagnostics or digital health. It's all about getting medicines to patients.
So tell me a little bit more. I think what you said was 3 years from human testing. Is that what you said?
Yeah, about 3 years. That's where the big value inflection occurs as well for us in life science VC is when you take an idea that looks good in test tubes and mice and actually manage to take it in the clinic and develop proof of concept that it actually can have efficacy and safety parameters in humans.
So can you tell me more about the origins of the sorts of companies or entrepreneurs that you're looking at?
Yeah, so as I said, there are really 3 basic models we use. The first is licensing academia. The second is licensing assets from biopharma, and that can be large or small biopharma. As you probably know, many of the very successful drugs on the market are things that companies have deprioritized, strategically out-licensed to others, and then those have been successfully developed, sometimes in the same indication that people had in mind, other times different indications. And the third model is what we call de novo drug discovery, where we really invest in a team of experienced drug discovery development professionals who want to pull together their team and begin to prosecute work. In academia, for example, we frequently encounter the situation where the founders believe that they're just about to go in the clinic, but in fact, they're not. And we have to look at it, and then we say, well, yeah, you're really more like 2 years from the clinic, but that's okay. because we, and particularly Desmond Patty, our principal, who's focused in this translational space his entire career for 30 years, we're pretty good at assessing that. And that determines the raise, the size of the raise, along with things like, can you contract the manufacturing of whatever it is? Or do you have to build internal manufacturing, process development, manufacturing, because it's a, let's say, cell-based therapy or a gene therapy, where the contract manufacturing organizations just aren't developed enough to do it for you. And then obviously that will result in a bigger Series A raise, a bigger check from us, more likelihood that we bring in another investor to write a big check alongside us, that sort of thing.
What mistakes are they making when they think that they're ready for clinical trials and you say, no, you're really 2 years out?
Well, I think, that what we see often from the academic centers is tremendously strong biology and early chemistry work, like screening, high throughput screening capabilities these days, and identifying, if you will, leads, and going through sort of hit to lead and identifying tool compounds that can be put into animal models. And what often is missing is really a lot of the assessment of whether these molecules are actually going to have the drug-like properties, for example, their metabolic profiles, half-life in humans, all that sort of thing, that would allow them to be clinical candidates that you would want to invest, $3-4 million in GMP manufacturing, GLP toxicology studies in a couple species. And when you put that lens from our experience onto a lot of these programs, the biology is great, the ideas are great, the chemistry is a starting point, but they're not as advanced as is believed by the founders. And this is not surprising because most of them don't have that deep industry experience of understanding how important things are, like drug-drug interaction potential, drug metabolism issues that can differ substantially. Mice diverged from us 65 million years ago in evolution, and they're not the model of humans. And these, a lot of the people that work in academia, they have their models and they're very focused on them, and they can sort of forget that there's an awful lot of work that has to be done to go from these tool compound mouse experiments that they consider to be sort of proof of concept to showing proof of concept in humans.
Do some universities do this better? Do some, do you wish that some of these labs were doing more of what you're talking about?
Yeah, I think that the universities in general are beginning to recognize that grant funding from NIH, and with a couple exceptions, there are some specialized grants that are designed to help entrepreneurs at academic centers sort of spin out their work into small biotechs, and that can be very successful if they do it right. But I think there's a real understanding now evolving at the medical center type universities that some seed funds within the university that can be dispersed to projects to get them a little bit further along and allow them to do things like pay a CRO to do these metabolic profiling experiments, to get crystallography performed, things of that sort. There can be arcane rules and regulations around how grant funds can be used. And we see this all the time, like you can't use a vendor outside the United States using NIH grant funding for some reason for some of these grants. And so they haven't worked with the best CRO that does crystallography in Germany, whatever, So these funds can be made available, and we've seen a number of the universities we work with set up these funds. It's often just a couple $100,000, but that's real money at this stage that we're talking about.
That makes a lot of sense. I have my siblings and my parents are all struggling to get NIH funding. So let me get into your background a little bit here. I think it's really relevant. You were running R&D at Amgen. How does Amgen think about early stage drug development and doing R&D versus acquisition?
Yeah, I would say Amgen is no different than what goes on across the industry in terms of large biopharma, in that there's a clear acknowledgement that if you look at the history of the industry and the trend line, more and more of the innovation come from venture-backed small company. And you can see this in terms of the proportion of the late stage visible pipeline at the companies that is derived from those sources has been steadily rising over the last 15 years. The number of independent biotech companies that actually launch products into the marketplace themselves is steadily rising. So less of the real earlier innovation is coming from the big biopharmas. In part, that's because they're consolidating and there's less of them, but also they have to spend a lot of their money on late stage development and marketed product support and safety surveillance and setting up new commercial operations with appropriate R&D support in different geographies around the world to exploit the emerging marketplace that's happening in places like China. And so all these things compete for early preclinical research dollars. Take as an example gene therapy or cell-based therapies for cancer. I mean, these are the two most obvious to point to recent big breakthrough kind of technologies in our field, right? You look at them and ask the question, how many of those, and there are hundreds and hundreds of them, right? How many of them were developed in large biopharma? To my knowledge, the answer for both modalities is 0.
Wow.
Okay. And if you think about that, what you realize is the dynamics that happen in any boardroom, in any of these companies, is you go in and say, hey, gene therapies coming to age. I want to build a gene therapy capability within the walls of, Pfizer, Amgen, what you pick your company. And why don't you give me an extra $1 billion, and over the next 4 years, I'll build gene therapy capability. And everyone will slap you on the back, say, you're a great head of R&D. I'm sure you can find $1 billion in your budget. And you'll get nothing, right? So that because they have to run a P&L and they have to make quarterly earnings, and the reimbursement agencies around the world and the CVS and Optum and so on in the US, they're all squeezing these companies really hard. And the profitability of new products is just not what it was for old products. So they're trying to manage all this. And meanwhile, they've got these very healthy balance sheets. And so then you come back to the boardroom 3 years later and you say, okay, look, we're going to fall behind the rest of the industry. We have to have a gene therapy capability. I want to purchase this gene therapy company for $5 billion. And this company will become Pfizer Thousand Oaks, and it'll do our gene therapy. And they have manufacturing and everything. And everyone will say, great, where do I sign? But they're generally acquiring that technology from venture-backed companies, either pre or post the companies being public. They can buy them before they go public or buy them after they go public. And it's always been that way to an extent, but what you're seeing is an enormous growth in that venture-backed component and less emphasis in large biopharma, at most of them, in really cutting edge innovation, because they kind of know they can buy it. Yep. And it's expensive to do. And that's the dynamic that's going on in the industry that makes it true that some of the most exciting, innovative work is happening in venture-based companies. And this was a large part of why I made the decision to move when I did. I want to be really clear. I'm not putting a finger on Amgen here when I describe this. This is a universal phenomenon.
Sure, of course. And what have you seen for these early stage companies that might be eyeing an acquisition by Amgen? What have you seen work, or maybe more importantly, what have you seen not work?
Yeah, great questions. I think one of the other mistakes that we see sometimes is that there's a decision made to hire a team. Now, I mean, of course, there's academic founders who can be invaluable and they can serve on the board or on the scientific advisory board and be invaluable to the company. I'm not talking about them. But what will happen sometimes is that the university will incorporate, which is in and of itself not a problem, but that company begins sometimes to get staffed with somebody who somebody knows, who has some kind of business MBA type background or what have you, and they get plugged into this job as the CEO, and they're not the right person to run the company because it's a science-based company and they just don't have the background experience, whatever. Because again, remember, these are very early companies where virtually every employee is going to be an R&D person who's trying to get something done, right? And it's very hard sometimes for people with just a business development, business MBA type background to even be able to relate to the scientists or understand the science and to lead that effort, nor is it very easy for them to know where this is all kind of going. So yes, there are people who can transcend that, but most of our CEOs have either, a PhD, MD, or both. And of course, as you might imagine, those kind of individuals with that phenotype tend to gravitate to work with us because we all have that background ourselves.
Mm-hmm. Right. And sorry, and I should just clarify who you all are. Obviously, we've talked about you. You're coming from head of R&D at Amgen, and you started Westlake with Beth Seidenberg, who's coming from Kleiner Perkins.
Yeah, so let me give a little history because it's a bit different than the typical life science venture group. In this case, yes, Beth worked in the industry at Merck, BMS, and Amgen, and we met at Merck more than 25 years ago. And we worked together there. We worked together at Amgen. And then after she was global head of development at Amgen, and then she moved to Kleiner. And that was about 15, 16 years ago now. I stayed on at Amgen, and so did Desmond Patty, our principal. Beth went on and created, I think she's now created 25 biotech companies. And she was the lead investor at Kleiner Perkins. She has a very similar background, physician scientist. And so we have an unusual set of backgrounds in that we're really operating executives from the industry who really lean in very hard to the projects, to the technology, the science, the medicine on the projects.
Wow. And you just said that Beth has created 25 companies. And I imagine some of that is the work that Westlake Bio has been doing to create companies. And some of that was when she was at Kleiner.
Yes. And between Kleiner and here. So we, in our first fund, we built 11 companies. And in the second fund, we've established 3. So that's 14. And then I think she did, 10 or 11 or something at Kleiner. So it's roughly 25. I'm not sure she even can keep track anymore. And most of those have been successful. The vast preponderance of them have gone on to exits. Between the 3 of us, Desmond, Beth, and I together, most things we look at, we've had some prior experience working in the field. And if we haven't, we often won't invest because, we want to work in areas where we can play to our strengths.
Can you double click on that a little bit? Which is I would say, another HR tech company. I've seen so many of those. Do you have the same thing where you're like, oh, prostate cancer, God, that's so overdone?
Yeah, well, the whole field of, immuno-oncology, and particularly immuno-oncology that's focused in hematologic malignancies has gotten a bit this way. I mean, you do get into situations where there's like 150 cell-based therapies directed at the same antigen, and really we're going to start up the 151st. So there are situations where you say I'm just not doing that. But in many cases, you say something like prostate cancer. Yes, I've worked on a lot of attempts to help people with prostate cancer, but most of them haven't really done enough. And there's still an enormous unmet medical need. And, that's the fundamentals along with people. So there's a timing issue for sure. You wouldn't have wanted to be investing in gene therapy 10 years ago. and you're probably not going to be wanting to do it 10 years from now. There are sort of times where the technologies, ripen and become tractable. There's nothing more frustrating practicing medicine than not having anything that you can do for patients. So you take an area like chronic pain, you'll very quickly exhaust your options in chronic pain. People can't tolerate long-term NSAIDs. You can't treat them chronically with opioids. You just run out of options really quickly. And if you have an unmet need like that is sort of one of the main reasons people seek medical help and the doctors don't have anything to offer, well, that may be a tough problem, but it's one worth trying to solve.
That makes a lot of sense. So are there companies that you'd like to be seeing a lot more of in your pipeline, or more of or less of?
Even in the areas where you look and you'd say, okay, autoimmune disorders like rheumatoid arthritis and psoriasis and those kind of things, they're well addressed. Well, not really. I mean, yes, we're a lot better off than we were when we were injecting gold into people when I was in medical school, right? But none of these drugs they're all immunosuppressants, and they're all fraught with all kinds of issues because of that. And at some point, it's going to be possible to, essentially reset the immune system so it no longer believes that self-antigens are foreign. If somebody brings, forward a technology like that, it'll be transformative. Beth always says, you have to be willing to sort of suspend disbelief and listen and think, because our job is to be thinking about what's going to be the therapeutic landscape, 5 to 10 years from now, because there's a long life cycle here. And we're open really to anything that can have a kind of, people use these words all the time: transformative, disruptive, it gets old. But that's actually what we're thinking about, right? This is one of the things I say all the time is this is incremental. It's just not transformative. It's nice. I like it, but it's incremental. And we just don't do that kind of stuff.
One of the big news stories out of biotech that hit my tech news was Google's AlphaFold and protein folding. Do you think that's truly transformative? Is it living up to all the hype? And do you see a big role for tech in biology, or how do you see that role?
Oh, yeah, absolutely. I mean, you don't want to overhype this kind of thing because, it'll have a huge impact, on one of the things that's a barrier that holds us back, that keeps us from being able to come up with, let's say, a small molecule to drug, a protein target, because we now can understand the structure of that protein much more easily, or there are the computational tools that allow you to see the motion that these molecules, they're not frozen in a crystal lattice the way we tend to think about them. They move. There's all kinds of things. And I saw this working in the genetics area when we acquired Decode when I was at Amgen. I mean, the kind of work they're able to do today simply wasn't possible until, Illumina developed the kind of sequencing capabilities. Sorry about the doorbell. The dog's going to start barking next. So I think there's absolutely no question. And the earlier you go into the drug discovery continuum, the more this is true, that these kind of technologies, different ways of imaging, different ways of analyzing, storing vast amounts of data, robotics, all of that stuff, they make a big difference. And I often, when I give this kind of lecture, I give sometimes around the golden age of biotechnology. I mean, one of the things I really stress is that a lot of the advances that are occurring that are making biology a more quantitative, more predictive science and more translatable into new therapies for people is the introduction of these non-biology technologies, whether it's robotics or X-ray, high throughput X-ray crystallography, or, these kind of things have profound implications. But at the same time, you can overhype the impact because we still deal with an enormous amount of uncertainty in things like choosing targets to pursue. You can work with all these tools in a very elegant manner on the wrong target.
Mm. But you'll invest in some of these tech platforms.
Well, no, generally not, because it's just not what we do. I mean, I have a hard enough time doing this work where I'm supposedly, like kind of a world expert, and I struggle every day to do the job because of the level of uncertainty. I mean, Robert Rubin said, and most people are more certain of everything than I am about anything. I subscribe to that camp, but we love to see the impact. And when we see that, hey, it was possible to drug this undruggable target because we were able to simulate a cryptic pocket in a computer, and we made a molecule and look, it actually works. We're like, okay, we'll take it from here with you.
That sounds great. Well, let me shift gears a little bit and talk about Los Angeles. You're here, Beth is here, your team is here in Los Angeles. What are you seeing, and how are we going to catalyze things in Los Angeles from your point of view?
Yeah, I mean, look, when Beth and I started talking about setting up Westlake, she had been commuting to the Bay Area for 14 years or something, but her primary home from Amgen days is still here, and that's how we've stayed so close. And I told her about my experiences where I had been asked by these universities to sit on these advisory committees about how can the university be more successful in commercializing their technology, right? How do we create companies locally where it creates job opportunities for our graduate students and our postdocs and all that. And what I would tell them, to their surprise often, is this area has tremendous academic institutions. I think it's number 3 in NIH funding in terms of a region in the United States. So that's not the problem. Lots of great people who could come in and be research scientists in the companies. We've got lots of industry experienced people from places like Allergan, Kite, Gilead now, of course, Amgen. And what's really missing, and the reason that the companies are not being built, is there's no venture group that focuses on, because many venture groups just invest in other people's A and B rounds and things. That's not going to help you. And I kept trying to say to them, you got to attract somehow, Atlas Ventures or the Column Group or somebody to set up offices down here in like Pasadena or whatever. And because it's a local activity that's very hands-on to build these companies. So when Beth and I started talking about the firm, and she started telling me, well, how overheated San Francisco and Boston have become, how crowded they are with Bay VCs, how the war for talent is resulting in constant turnover of people, including R&D staff, which is really bad, to have high turnover in that group. We just started talking about the fact that we thought there was a tremendous opportunity to build companies here, that basically everything was here. Grassroots support from the government. I mean, you can't imagine how helpful the city of Thousand Oaks or LA County have been. And we didn't know it at the time, but Alexandria Realty, who are headquartered here in Pasadena, they've been wanting for 20 years to do their magic in this area of building biotech incubators and that kind of thing. But there just wasn't enough company formation going on to warrant it. And now they're doing that. We have 5 of our companies right now in a first Alexandria facility here close to the Amgen campus. And they're going to build more, and it's really taking off. The characteristics here are as favorable as I think they were in places like Boston and San Francisco for a biotech hub. I mean, Beth and I both watched those ecosystems evolve. And, the one thing that happened in those places that was really seminal was big companies started buying small companies and establishing their presence. So Gilead, of course, bought Kite in Santa Monica, but what'll happen next is a company, I don't know, Atara or one of the companies that's local here, will get bought by, Novartis or pick your big biotech. And then there'll be a Novartis sign here. Our biggest challenge of doing the company formation work here was just ready, turnkey, available laboratory space. And now that's been largely solved by Alexandria.
That's really exciting. And so you've gone, your first fund, was it a 320 million first fund? So now you're working on this 500 million. It's really exciting for the ecosystem. Let me ask you maybe one or two final questions. What's different than you expected being a VC versus doing R&D?
Yeah, I think the first thing was just sort of how you actually go about doing all of the financial and mechanical aspects of actually putting together companies, licensing technology out of universities, figuring out the cap table, all of this stuff. And to figure that all out at 56, again, after I'd come from being kind of, the expert guy in what I was doing before, and now I'm completely clueless about what I don't even know what I don't know. That was uncomfortable, but I never looked back because I knew in the long run it would be great. And it is, you can create one of these companies and 2 years later, the company's doing a major deal with a big biopharma, and before you know it, then they're talking about becoming a public company. It's kind of magic. It's really quite amazing that sort of 0 to 1. Well, you can read books about that, but to actually be involved in that value creation and employing all these great people and all that, I found that that's been better than I imagined.
That's fantastic. Well, I think your work and your mission is really inspiring. I appreciate what you guys are doing to catalyze the ecosystem here in LA.
Likewise, you're doing the same, of course, but it's been a pleasure talking to you.